Behind the Breakthroughs, conversations with dementia research pioneers on ISTAART Research Retrospectives.
Hello, my name is Claire Sexton, and it's my pleasure to be chatting today with Dr. Alison Goate and to go on this research retrospective of her incredible career. Alison, welcome.
Thank you, Claire.
So today we will be discussing two of your landmark papers. The first, segregation of a missense mutation in the amyloid precursor protein gene with familial Alzheimer's disease, was published in Nature in 1991. And the second, integration of Alzheimer's disease genetics and myeloid genomics identifies disease risk regulatory elements and genes, was published in Nature Communications in 2021. So firstly, can you believe that 30 years passed between the publication of those two papers?
In some ways, yes, but in some ways, no, right? The time went by very rapidly, and frustrating that we are only now having therapeutics that are focused on amyloid in the clinic when the mutations were found 30 years ago. It's that's discouraging to think it took so long.
It took a long time, but
yes.
We're there.
We are there, yes.
So first, if we go back to your first publication in 1991 that we'll be discussing, you were at that time at St. Mary's Hospital Medical School in London. Would you be able to set the scene for us then?
Yeah, sure. So I started working with John Hardy in 1987 as a senior postdoc. And then in 1989, I got a Royal Society University Research Fellowship. And so I started developing my own independent projects. So, by 1991, there was sort of some of my own projects and this project that I started with John on the familial Alzheimer's disease. And sort of, I got married in 1987, had my first child in 1991. So it was a busy 4 years. And obviously the fact that the paper came out at that time when I was pregnant was obviously very, very busy year, 1991.
And you mentioned that then you were working on familial Alzheimer's disease.
Right.
Would you be able to share with our listeners a bit more about what familial Alzheimer's is? What was known about the causes, back in 1991 and what motivated you to...
So for genetic studies, these familial forms of Alzheimer's disease were really important. So they're pretty rare, but there's a clear mode of inheritance. So if you have an affected parent with early onset familial Alzheimer's disease, you are at 50% risk of developing disease yourself. And so for geneticists, then that clear mode of inheritance means that you can use familial approaches to study the disease. So what John and I did was, with Martin Rosser, collect families in the UK where there were multiple cases of Alzheimer's disease in people below the age of 60. And it was these families that we used for the study in that Nature paper.
And by, studies involving these families, then you were able to discover that there was a mutation in the APP gene in two unrelated families with familial Alzheimer's. And this was the very first suggestion, if I'm right, that some cases of the disease could be caused by mutations in APP. And you've described, how this was, changed the trajectory of your career.
Yeah. So actually, back in 1987, there were several papers published that said that there weren't mutations in the amyloid precursor protein gene. And so people made a false conclusion at that time, everyone thought there was going to be one gene that caused these familial forms of Alzheimer's disease. And therefore, if you could exclude a mutation in APP in one family, you excluded that idea in all families. And so that kind of set us back. That was actually the time I joined John's lab in 1987. And so then we started using sort of a pure reverse genetics approach to identify the region on chromosome 21 where we had linkage already, but to try and narrow it down and find which gene it was. And so we were quite fortunate that among the families we were studying, we had a family that we really couldn't rule out APP. There was a strong signal just above the APP gene. And so we went back and sequenced APP and then found a mutation in that family. And, so that really changed everything, because then people started looking at individual families. And obviously we know now there are quite a few families that have mutations in APP. But for that period of 4 years, everyone assumed that there weren't mutations in APP. So, it means always be, mindful and careful when you're doing your studies, right? Because that was a false conclusion that sent everybody in the wrong direction for several years. But that was really a groundbreaking study that allowed us in the field to develop animal models, to develop cell models, which we never had before. To study the disease in the late 80s, you basically used brain tissue from, people who died of Alzheimer's disease. So you're looking at the end stage of disease. Finding the mutations allows you to create cells and animal models that carry these mutations, and they don't produce Alzheimer's disease, but they mimic some of the molecular processes. And at least in the animals, you get amyloid deposition. And so it allowed people to study that process and ultimately to develop therapies, obviously.
Yeah, and at the time, did you, have an inkling of how big this finding was?
I think yes and no, right? I mean, I definitely, knowing that we had a cause of disease, but not really fully understanding at the time, what proportion of cases might be affected by it or how useful any of the models might be. I mean, obviously today, 30 years later, we're still using, the amyloid models that were made in the 90s to study amyloid deposition in mice. So, and it certainly, for me personally, it definitely changed the trajectory of my career. There were times before we found the mutation that I had thought that I might need to get out of science, because I hadn't published very much. I didn't think I was particularly competitive to get a faculty position. And so I was trying to think what I might do next. And obviously this paper changed all of that. So for me, it was a huge deal.
Gosh, that's amazing. So it was really, a springboard for you and for the field.
Yes. Yeah.
And now if we fast forward 30 years now, the focus of your work has evolved from, discovering these kind of rare causal genes to deciphering more common genetic risk factors. And advances in methods were crucial, it seems, to this evolution, that there was, GWAS studies by you and many others that contributed to a shift in focus for the field, looking beyond just amyloid and also tau, but understanding the complexity of the disease and, placing a spotlight on the immune system in particular. And this paper that we're going to discuss in 2021 in Nature Communications employed such a variety of different measures to then connect genetic risk with Alzheimer's with changes in the microglial gene expression. And, could you share a bit about how, the methods in genetic research changed over that time and the impact that it's had?
So you're right that really GWAS wasn't possible before. The earliest GWAS studies were in about 2006, and really big studies didn't happen until 2010 or around there. And I think, so the revolution that happened was basically having sort of a chip that you could look at DNA variation in millions of places in the genome simultaneously in one individual, but then you can multiplex that and do hundreds, thousands, tens of thousands of people. And we were doing that all by hand. So in 1991, a polymerase chain reaction was new, right? A PCR machine was 3 water baths, different temperatures, and you move things one water bath to another, right? So obviously you're not going to look at hundreds of thousands of variants or even thousands of people if that's some kind of approach. So really it was technological advances that made things happen. I think the other thing that's really different between the two eras is it was a race in 1991, right? That you could only win that race if you had families in your collection that carried a mutation in APP, right? So some people didn't have any. But also the glory went to the people who got there first, right? Now, human genetics for complex diseases is a collaborative process, right? That nobody can do this alone. We all work together. It's team science. And so I think that's a huge difference in the way we approach our work now compared with then.
And I'd love to unpack that a bit more. Yeah. What does team science mean to you?
I think working with groups and teams around the world with a common cause to solve a problem, right? And people in that team come with different expertise and view the problem from different angles. And I think that those things make doing this kind of research exciting and invigorating. And to me, it's much more satisfying to be involved in that kind of team science. I mean, for us, in 1991, the team was our individual lab, whereas I would say now, it's people working on genetics of Alzheimer's disease all around the world. That's the larger team, and that we need each other in order to accomplish our goals.
And that approach then, allows you to, publish those types of papers like your 2021 paper. Where do you think, where can you see the impact of that paper having then in the coming years?
Yeah, I mean, I would say, so it's not just that paper, but I would say over the last maybe just under 10 years, there's been a realization of the importance of the immune system in Alzheimer's disease that wasn't previously appreciated, right? And so, I can remember going to Alzheimer meetings where, discussion of the role of microglia in Alzheimer's disease was at the last session on the last day, and most people were already left to go home, right? Whereas now it's the plenary on day one. And so I think that shows you how much the field has changed its thinking. And it was really started, I think, with the discovery of TREM2. But, since then, it's become clear through our work and other people's work that the majority of genetic risk for late onset Alzheimer's disease is found in microglial expressed genes. And so that, I think, is really important for moving the field beyond amyloid and tau, right? I mean, it's clear that we desperately need novel targets and novel approaches. Most other complex diseases, cancer, heart disease, we have lots of different ways of trying to treat those diseases. And right now in Alzheimer's disease, we do not. And so that's really important to be able to broaden our horizons in that respect. So
completely. And so I think you've brought us up to speed with how the field has changed in its focus, how the methods has changed over this time, how the nature of collaborations has changed. But I wanted to touch upon how your environment changed too over this time, going from working in London to then living in New York City. So what type of, impact did that have?
So I would say part of the reason that I moved was that in the early 90s, research funding in Alzheimer's disease was not all that great in the UK. And when I moved to the US, I discovered the Alzheimer's Disease Research Center programs by NIH. And I think that that's been a tremendous program for supporting Alzheimer's research in sort of generating well-characterized cohorts for study. So from the research perspective, definitely the move was related to being able to do the best research that I can do. Living in Manhattan, I love living in Manhattan at this stage in my life. I don't think I could have brought my kids up there. I did that in St. Louis, and it was a good place to bring up my kids. I think that now they probably feel more American than they do British, but they had to carry both passports. So, yeah, I mean, I think having brought up my family in the US, that's probably sort of a big difference. All of my extended family still lives in the UK. So,
but the US is home now, would you say?
Yeah, it is. I mean, certainly while I'm still working, I really can't imagine moving back. Actually, I don't really want to move again. Moving is disruptive. And so, no, I've made my home in New York at this point.
So on that note, I would like to thank you, Alison, for sharing such important insights over 30 years of a stellar scientific career. And here's to another 30.
We'll see. Yes. Thank you very much, Claire. It was fun talking with you.
Behind the Breakthroughs, conversations with dementia research pioneers on ISTAART Research Retrospectives.